This video correlates with Mendelian Genetics Part 4, and that is your notes page 26B. Okay, so we know that genes are located on chromosomes. They're just sections of DNA on the chromosomes. And there are two types of chromosomes that are associated with humans. We've looked at a picture of a karyotype similar to this before, but those two major kinds of chromosomes are called autosomes and sex chromosomes. So if we look on here, I've told you before that human chromosomes are arranged in order of size from the largest to the smallest, and then they put the sex chromosomes at the end. So chromosomes right here, numbers 1 through 22, are considered autosomes. Okay, so if up above autosomes on your notes, if you will put number 1 through 22, numbers 1 through 22. And then if up above sex chromosomes, you will put 23rd pair. Those are going to be your sex chromosomes. So that's where we get the XX or the XY. Those are your sex chromosomes. And of course, chromosomes are inherited from the parents. Now here we know that this is a male because they have an X chromosome there and a Y chromosome there. So that's their 23rd pair. As opposed to this next slide, this is going to be a female. And we know that this is a female because she has two X chromosomes, but no Y chromosomes, okay? So males are XY, females are XX. Now, when it mentions there about linked genes, these are genes that are usually inherited together as a unit because they're found on the same chromosome and they're found really close together. So they're usually going to be on the autosomes, that numbers 1 through 22. I want you to write out an example on there, on your notes, out beside where you have linked genes. So to give you an example of that, if you'll write this down, the example would be red hair and freckles. The genes that control those two things are typically present on the same, they are present on the same chromosome, and their locus or their address on that gene, they're really close together. And so typically when they get inherited as a unit. It'll include both genes instead of just one, and that is why individuals with red hair often have freckles as well, because those genes are typically linked together. Now there's a gentleman called Alfred Sturtevant, and he is the one he's listed on your notes there. He was the pioneer of what's called genetic mapping, so identifying the loci of the genes. He used crossover rates in prophase 1 to determine the loci on the chromosomes. And when he got through with that, he called this a linkage map. So the smaller the rate, the closer that they are in relation to each other. Okay, and the larger the rate, the farther apart they are from each other. So in this case, when we're looking here at these, for example, these would be closer together than, say, this one would, that would be farther apart. So he is the one that just kind of addressed or gave them the address on the gene of each of these specific traits. Sex-linked genes are those that are on that 23rd pair of chromosomes, your sex chromosomes. So I want you to put there pair number 23, okay, is where our sex-linked genes are going to occur. Now, 95% of the time, it mainly refers to the X chromosome. So you can think X, it says on your notes, when it is sex-linked because there's an X there. And that's just because both of the sexes have at least one X chromosome in their genome. So it's going to be the one that's most commonly affected. Y chromosomes, females don't have those. So it's going to be more often that it would be X-linked. So XX, it tells you that's female, and they're called homologous. XY as males, and they are heterologous. All right, so sex chromosomes don't undergo a whole lot of crossover during prophase 1 of meiosis. And the sex of an organism is actually determined at conception. When the egg is fertilized by the sperm, you're either going to get the female, the mom is only going to donate an X chromosome. That is all that females produce is X chromosomes. So the female is just going to donate an X, and it's up to the dad as to whether he will donate an X or a Y chromosome. Males make both X and Y. So if the dad gives an X, if that sperm cell contains an X chromosome, then the offspring will be female. If that sperm cell contains a Y chromosome, then they will be male, because again, the mother can only give an X. So dad gives an X, it's XX, as in the case of this karyotype, and it will be female. Or the dad can give a Y, and so it'll be X from mom, Y from dad. It'll be XY, which would be a male. Now, your gender is decided at conception, but as far as development, everyone actually starts out female. In about 2 months of age after conception, the Y chromosome, if it is present, if it's a male, then there's an SRY gene that gets turned on in order to turn on testosterone. And then the male will finish developing and the testes will descend and all of that. And he'll develop those male characteristics. And then that gene, the testosterone, gets turned off and then turns back on during puberty. So that it can make the secondary sexual characteristics like the deepening of the voice and hair distribution and all of that. So when we're talking about sex-linked disorders, genetic disorders, these are going to look a little bit different as far as the way that they are presented in a Punnett square, because they are just carried. Sex-linked means that they're just carried on this 23rd pair of chromosomes. And again, just want to remind you here that, that you've got an XY for males, okay? And that's what that 23rd pair is going to look like. And you're getting one of those cells, one from each parent for the sperm and the eggs. All right, so let's look at color blindness. If you have a sex-linked genetic disorder, there is no cure because the problem is actually in the DNA. So in color blindness, the problem comes in with a faulty gene. It's a recessive trait on the X chromosome for making a particular type of light wavelength absorbing protein. So it's problem making a protein. Again, remember genes or DNA, contains the instructions for making proteins. So they fail to make that protein that goes in the cones and the retina of the eye. And so the most common type is red-green color blindness. The red and the green just appear gray. Here's actually a test, a very simplified test. They'll do, it'll be more elaborate than this. But if you're reading these dot charts, people that have red and green color blindness often cannot read these. They will just see a series of dots. They're unable to see the numbers that are present there inside those circles. So anyway, just a little picture to kind of show you how they diagnose that. Okay, so when we're looking at the pedigree chart, notice what gender is mostly affected by color blindness. All right, now remember, if they are affected with a particular trait, they are going to be in a solid color. If it's half and half, that shows you a carrier. All right, our squares are males, circles are females. So clearly you can see with the color blindness, it's primarily males. Every once in a while, we have on this particular pedigree chart, we have one female that has color blindness. But we have 1, 2, 3, 4, 5, 6 males that have color blindness, and we have some female carriers. So let me show you why that is the case. Okay, so looking at that pedigree chart, we had a father that had color blindness, and we had a mother that did not have color blindness. All right, well, when you're doing a sex-linked Punnett square, you know that you have a dad that is XY. You have a mom that is XX. Okay? So that is the way we actually set up a sex-linked Punnett square. All right, now it tells you in your notes there that color blindness is due to a recessive trait on the X chromosome for making a certain kind of protein. So because dad has color blindness, we're going to put on his X chromosome, we're going to put that faulty recessive trait. So we're going to use a lowercase c for color blindness. Capital C we're going to use to represent somebody who is able to see color. So mom does not have the trait and she is not a carrier. So we're going to put her as having normal color vision. All right, when you bring these X's across and down, all right, we've got mom gave us the normal X there. Dad and mom gave us, from the dad, we're getting this X there with the faulty color blindness thing there. Colorblind trait. All right, so you're bringing those down, and then we wind up getting this across. All right, now if we flip back to this pedigree here in just a second, notice our offspring. One of the cool things about sex-linked offspring is it divides them into genders for you. Okay, so these right here are XX, so those are females. So you have two females that are carriers. All of your females are carriers, okay? Because on their X's, they're carrying a normal color gene and an abnormal color or color blindness gene, okay? So they have one big C, one little c. So they're heterozygous. They are carriers. Now look here at your males. These are XY. Okay, look at 100% are basically normal males. They have no color blindness. Okay? Even though the father was colorblind. Now let's go back up and look at that. And if you'll look at the colorblind father and the normal colored vision mother, okay, up here at the top, look at their offspring. Every one of their daughters is a carrier of color blindness, but every one of their sons has normal colored vision, just like we said, okay? But let's look here at this couple, okay? They're going to have offspring. Mom is a carrier for color blindness. Dad doesn't have color blindness, okay? So let's do a Punnett square for that. So we said mom is a carrier. So that means she has one normal gene and one faulty gene. Dad doesn't have color blindness at all. Okay, so he's XY. And remember, the Y is not affected. It's on the X chromosome. So when you fill in your Punnett square, this is what you wind up getting. All right, look at our females. 50%, here's our females right here. 50% of them, or half, 1 out of 2, is normal vision, normal colored vision. However, 50% of our females are carriers for color blindness. All right? Now, when you look at the males who are XY, you've got 50% that have normal vision. The first one up there, right there. But 50% are colorblind. All right, and the reason, and this is why it is much more common in males than in females, sex-linked traits, because if you look here at this, this genotype, males only have one X. So they don't, if you're talking about complete dominance, they don't have any other trait that will dominate over that colorblind trait to give them normal color vision. If they have a colorblind trait on that X, they are going to have that trait. They are going to have color blindness because the Y doesn't have anything on it to override it. It's a completely different chromosome. Okay, now if you look here at this female, in order for a female to be colorblind, she's got to get that recessive trait from both parents. Otherwise, that dominant just dominates over the little guy there. She doesn't have colorblindness because she has another X to override the colorblind trait. So we have said here that we would have 50% of our females would be normal and 50% would be carriers. We've got 50% of the males will be normal and 50% would actually have color blindness. So looking at our pedigree chart, let's see if that actually panned out. Now we were looking at this couple, okay? So we're looking at these children. All right, so we said that there's 50% chance that the females could be normal, and there we do have a normal female. 50% that they could be carriers, and there we see a carrier. 50% chance that a male could be normal, and there he is, and 50% chance that a male would have color blindness. And it checks out just like the Punnett square indicates that it probably would. Hemophilia is another example of a trait that is carried on the sex chromosomes. Now, they are not able to make anti-hemolytic factor, which causes blood clotting. So hemophilia, philia means to love, hemo is blood. Hemophilia actually means to love bleeding. They are unable to clot, okay, to make blood clots. So they could potentially bleed to death. Now, you may have learned in history about what they call the royal blue bloods of Europe, and it was a Russian family. Anastasia was one of the, I guess, the famous people out of that family. But because of they wanted to keep the crown in their family, so they started doing a lot of inbreeding. And this resulted in the development of a hemophilia trait. Many of these individuals bled to death, even from simple, something that looked just like a bruise could cause them to bleed to death and lose their life. And that is exactly what happened. They were having a lot of their heirs to the throne were dying of hemophilia. They were bleeding out. And so there is no cure for this. What I say cure, it doesn't stop this from happening, but what we do have is they can take anti-hemolytic factor, and if they get hurt or something that causes bleeding, then they actually take an injection of the anti-hemolytic factors to stop the bleeding, and it can be a very serious issue if they don't do that. And in looking at this pedigree chart where you have Alice here, who is a carrier for hemophilia, so I have a picture, or I've got her genotype here. Okay, so she's a carrier, so she's heterozygous. Hemophilia is also a recessive trait. And then the father, Louis, does not have hemophilia, and so we use that capital H to represent individuals that have normal bleeding and do not have hemophilia. The lowercase h there represents hemophilia. So when they had children together, if you look at the females, they could have a 50% chance of having a child that was not a carrier and did not have hemophilia. So there you see that they have daughters. 2 daughters that do not carry the trait for hemophilia. But they also had a 50% chance of having a daughter that did, and they did. They had a daughter that was a carrier. Then you have, in looking at the males, this is supposed to be a capital H there. It's kind of hard to read. But anyway, they have a 50% chance of having sons that do not have hemophilia, and they do have Henry, who does not have hemophilia, and then they have a 50% chance of having a son that does indeed have hemophilia, and that is exactly what happened there with with Fred. He has hemophilia. And actually, I made a mistake there. Henry is not their kid. Henry married Irene. Okay, so let me retrace that here. It was, they have, see if we go all the way over and erase my marks I've done there. Okay, so all of these are their kids. We've got 3 daughters there that do not have hemophilia and that are not carriers. And then we have 2 daughters that are carriers. And we have a son that actually has hemophilia. These 2, Henry and Nicholas, the second of Russia, are actually married in. They married Irene and Alex. So you can see it's carried on the X chromosome, but that is why it is more common in males, because they only have one X. So females, if they happen to get a trait like that on the sex-linked traits, then they typically are just carriers rather than actually having it, because most of the time they're going to have an X that's dominant that's going to carry over and kind of override that one.